Hello students, welcome back to the biology class. So in the last class we started the new chapter, that is a new concept also for you, that is 11th chapter, biotechnology and the principles of and processes dealing with the biotechnology. We learned that this term was coined by the Hungarian engineer Karl Ehricke in 1971, and biotechnology is basically it involves the techniques of using the living organism, could be microorganism or multicellular plants, or otherwise the animal cells, or otherwise using their components or enzymes from organisms to produce products as well as processes which are required for serving the life, the daily life of the human being. So this concept was introduced in detail by the genetic engineering father, who is also considered as the father of genetic engineering, Paul Berg, in 1972. And he is the scientist who is given credit for the birth of biotechnology because he was the first scientist who introduced a piece of DNA from SV40. SV stands for the simian virus, a virus which attacks the primates a lot. So this simian virus SV40 gene was introduced with the help of the lambda phage into a bacterium. So this concept was introduced by him for the first time, and we learned in the last class the definition which was introduced as per the NCERT. I'm reading from the European Federation of Biotechnology has given the definition of biotechnology that encompasses both the traditional view as well as the modern view, and we learned that the definition is that integration of natural science and organisms, cells, parts of these cells as well as the molecules found in these cells for the product as well as for service. We learned that the recombinant technology came into existence because of Paul Berg, and then it was further the next mile step after Paul Berg's experiment was the Cohen and Boer experiment, which is explained even in the NCERT. We learned that biotechnology is use of any type of technology or technique in the living sphere so that we can serve the human life better, and that's why there are two methods of biotechnology, old and the modern. We learned in the last class that the modern biotechnology deals with the genetic engineering, and the old biotechnology is the traditional concepts of techniques, for example alcohol formation, curd formation. Even the concept of plant breeding and animal breeding are the concepts under the old or traditional biotechnology. So in the old or traditional biotechnology, we only restrict ourselves to the natural capabilities of organisms. So we do not manipulate their genetic material. We only take benefit out from whatever the organism is capable of doing to its maximum. So the problem with this method is that we cannot bring novel characters or more characters or something new into the characters of the organism. Like for example, we all learned in the last class that the plant breeding and animal breeding, it involves the transfer of the gametes from one individual to the other individual. But when we are transferring the gametes in the form of either pollen grains or the sperm, we are only paying attention to one or two characteristics which are of desire, but along with the desirable characters, even undesirable characters are also transferred with the help of the plant breeding and animal breeding. So this problem is overcome by the modern biotechnology, which is also called as the genetic engineering, because in this concept we manipulate the genetic material and we only transfer the desired gene or a set of desired genes. So there is no transfer of undesirable characters. So this concept is far more better when compared with the traditional method of biotechnology. Now this genetic engineering came into existence because scientists discovered something called as plasmid. They understood that it is able to replicate along with as well as independent of the chromosomal DNA. And then the discovery of restriction enzyme, that how nicely they are able to read a particular stretch of DNA and cut at a particular place only. And then the enzyme which is the DNA ligase. All these discoveries led to the development of today's genetic engineering or the modern biotechnology. We learned that during this concept there are two enzymes which are most important, that is the restriction enzyme, which is also called as the molecular scissor or biological scissor, and the second enzyme is DNA ligase enzyme, which is also called as the molecular glue. We learned that in the concept of genetic engineering or in the biotechnology of modern type, there are two things which we have to develop: genetically engineering the organism and then creating the chemicals or chemical engineering so that conditions could be made favorable for the genetically modified organism, or GMO, genetically modified organism, may it be prokaryote or eukaryote, to grow so that the product of desire could be prepared by them to their maximum amount. We started the topic of conceptual development of principles of genetic engineering. And we talked about plasmid is present in bacteria, which undergoes replication along with as well as independent of the chromosomal DNA. And we talked about restriction endonuclease enzyme.